Beta oxidation of fatty acid. Fatty acid oxidation occur in starvation and diabetes. First question. Yes, starvation and diabetes. Beta oxidation of fatty acid. You will remember, starvation, there is no glucose to produce energy. Cell will utilize fatty acid to produce energy, beta oxidation of fatty acid. In diabetes, blood glucose is high but will not enter the cell. Cell will think there is no glucose. In fact, there is no glucose at the level of cell. Cell will utilize fatty acid to produce energy by beta oxidation of fatty acid. So beta oxidation occur in starvation and diabetes. Gluconeogenesis also occur. Gluconeogenesis also occur in starvation and diabetes. In starvation, there is no glucose. Liver will try to maintain blood glucose by gluconeogenesis. Why liver is synthesizing glucose? Because you know most vital organ, brain, utilize glucose in fed state, fasting state. Even in starvation, brain try to utilize glucose, but it can utilize ketone bodies also. That's why in starvation, liver try to synthesize ketone bodies, ketoacidosis. Starvation ketoacidosis. Okay, diabetic ketoacidosis. So ketoacidosis occur in starvation diabetes, gluconeogenesis occur in starvation and diabetes, beta oxidation occur in starvation and diabetes. There is close relation between these three pathways. Okay, now beta oxidation of fatty acid occur in mitochondria. Suppose there is a fatty acid. Suppose this is fatty acid. Fatty acid, first of all, this fatty acid is activated and it is converted to acyl coenzyme by acyl coenzyme synthetase. Synthetase. You all know synthetase and synthase. What is the difference? Synthetase ATP used. You can easily remember synthetase. Even in the pronunciation you require a little bit more energy, so synthetase ATP is used. In fact, 2 ATP used. If I take palmitic acid, how many carbon atoms are there? 16 carbon atoms. Yes, how to remember? 15. Yes, 1 2 3 4 5 15, and the palm is 16. So palmitic is 16 carbon fatty acid. Now we want to produce energy from palmitic acid. First of all, this palmitic acid is activated. In this, 2 ATPs are used. Now acetyl coenzyme, not acetyl, acyl coenzyme want to enter mitochondria, but you know inner mitochondrial membrane don't allow few compounds. In the same way, fatty acid will not be able to cross inner mitochondrial membrane. It will take help of car of James Bond. It will take help of car of James Bond, carnitine. Carnitine. Okay. So question is asked, fatty acid enter the mitochondria with the help of carnitine, a compound other than carnitine. It also requires three enzyme, CPT1, CPT second, and translocase. And translocase. So with the help of these four things, carnitine, CPT1, CPT2, and translocase, fatty acid enter the mitochondria. If any one of these is not there, not functioning, you can't do transfer fatty acid from cytosol to mitochondria, but beta oxidation occur in mitochondria. So if you want to inhibit beta oxidation, you can simply inhibit by inhibiting this CPT1. And you all know malonyl coenzyme is inhibitor of CPT1. I discussed that malonyl coenzyme is having 3 carbon. Yes, when fatty acid is being synthesized, in fatty acid synthesis, acetyl coenzyme is converted to malonyl coenzyme. Malonyl coenzyme inhibits CPT1. You all know fatty acid synthesis occur in fed state. Beta oxidation occur in fatty acid synthesis occur in fed state. Beta oxidation occur in starvation. So in fat state, is there any need of beta oxidation of fatty acid? No. In fat state, we are having sufficient amount of glucose to produce energy. There is no need of beta oxidation of fatty acid. That's the reason that when fatty acid is being synthesized, this malonyl coenzyme is being synthesized. This will inhibit CPT1. When this CPT1 will be inhibited, this fatty acid will not enter the mitochondria, and when it will not enter the mitochondria, beta oxidation will not occur. So question is asked, inhibition of beta oxidation of fatty acid is done by CPT1 inhibition by malonyl coenzyme in fat state. Okay, yes. Now fatty acid entered the mitochondria. Recently asked question on this carnitine INICET. Yes, neat. Now this is palmitic acid. Beta oxidation means a cut between C2 and C3. C2 and C3. Yes, beta oxidation means a cut between C2 and C3. This cut is done by four enzyme: dehydrogenase, hydratase, dehydrogenase, and thiolase. Even if you don't remember so much, simply remember, yes, beta oxidation and fatty acid synthesis are two opposite pathway. I have already told you, fatty acid synthesis and beta oxidation are two opposite pathway. These are regulated in such a way, when one is activated, other is kept in inhibited state. I just explained, I told you, in the form of this insulin. Insulin activate fatty acid synthesis. At the same time, it inhibit beta oxidation of fatty acid. One way to regulate fatty acid synthesis and beta oxidation is by hormonal level. Another way is by malonyl coenzyme. When fatty acid is being synthesized, malonyl coenzyme is being synthesized, it will inhibit CPT1. Now, fatty acid synthesis and beta oxidation are two opposite pathway till we know. Yes, and so their enzyme should be opposite. Their enzyme should be opposite. Although not every time this concept is followed, but in this fatty acid synthesis, I told you the name of enzyme is reductase, hydratase, reductase. While the name of enzyme in beta oxidation is dehydrogenase, hydratase, dehydrogenase. And in complete biochemistry, wherever dehydrogenase is there, removal of hydrogen is taken up by FAD. First dehydrogenase, hydrogen is taken by FAD to form FADH, and FADH will produce 1.5 ATP. Dehydrogenase, second dehydrogenase produce NADH, which will produce 2.5 ATP. So 1.5, 2.5, total 4 ATP. So this bond is cut between C2 and C3, and by this cutting, yes, you know, dehydrogenase, hydratase, dehydrogenase, opposite to this fatty acid synthesis is, yes, reductase, hydratase, reductase. Question has been asked many times. Okay, now this two carbon unit will be released as acetyl coenzyme. This acetyl coenzyme will enter Krebs cycle, and I told you one acetyl coenzyme enter Krebs cycle to produce 10 ATPs. Okay, so palmitic acid is having 16 carbon divided by 2, 8 acetyl coenzyme. So I can say, if palmitic acid is completely oxidized, 16 carbon compound will give rise to 8 acetyl coenzyme, and 1 acetyl coenzyme enter Krebs cycle to produce 10 ATP. So 8 acetyl coenzyme will produce 80 ATPs. Okay, now these 80 ATPs by bit cutting, and you know, one cut produce, yes, one FADH and one NADH equivalent to 4 ATP. So to produce 8 acetyl coenzyme, you require 7 cut, 7 cleavage, 7 cleavage. Yes, and one cleavage produce 4 ATP, 7 into 4, 28 ATP. Yes, sometimes this calculation is asked. Yes, 80, 28, 108 ATPs are produced, but you know, 2 ATP were used in the activation of fatty acid. 2 ATP were used in the activation of fatty acid, so net gain would be 106 ATP. So complete oxidation of palmitic acid produce 106 ATP. Question, palmitic acid is having 16 carbon. Yes, palmitic acid is having 16 carbon. If I write 16 like this, Okay, and if I put one oxygen here, 106 oxidation, so net gain 106. This is mnemonic palmitic acid oxidation, palmic palm 16 carbon, and oxidation O0 looks like 0, 106 net gain mnemonic. Okay, so first function of beta oxidation is to produce energy that can be produced by complete oxidation. Yes, suppose a person goes into starvation, there is no glucose to produce energy. Liver cell took one palmitic acid, cut, 7 cleavage, 8 acetyl coenzyme, Krebs cycle, energy requirement of liver cell is fulfilled. Now liver cell will think of brain. If I don't synthesize glucose, brain will die, because even in starvation, brain prefer to utilize glucose. So liver cell will take one more palmitic acid, cut, 7 cleavage, 8 acetyl coenzyme. All these 8 acetyl coenzyme or acetyl coenzyme will activate pyruvate carboxylase, that is rate limiting enzyme. Question is asked, gluconeogenesis, rate limiting enzyme is pyruvate carboxylase, which is activated by acetyl coenzyme. In starvation, there is gluconeogenesis which occur in mitochondria. In starvation, beta oxidation occur, and this cleavage produce acetyl coenzyme which activate gluconeogenesis. So one function of beta oxidation is to produce energy. Second function is to produce acetyl coenzyme to activate gluconeogenesis. You all know there is starvation ketoacidosis, and when you look at the pathway of ketone body synthesis, I write here ketone body synthesis. Two acetyl coenzyme join to form acetoacetyl coenzyme, then HMG coenzyme, then acetoacetate. Question is asked, first ketone body to be synthesized is acetoacetate. Most common ketone body present in blood, yes, beta hydroxybutyrate. Question, B मतलब बहुत सारी, big amount, beta hydroxy butyrate. Okay, ketone body synthesis occur in mitochondria, beta oxidation occur in mitochondria, gluconeogenesis occur in mitochondria. All these, yes, gluconeogenesis start in mitochondria, and all these three pathways occur in starvation. So there is relation, means whenever beta oxidation occur, acetyl coenzyme enter Krebs cycle to produce energy. When energy requirement is fulfilled, then liver synthesize glucose for brain by gluconeogenesis. And when you know that glucose is utilized by each and every cell in our body, that's why brain is, you know, brain is very vital, and you know survival of brain is essential. Suppose liver synthesize few molecules of glucose, every each and every cell will utilize glucose. And all, suppose liver made glucose by gluconeogenesis and released in blood, each and every cell will take that glucose, and till this glucose will reach to brain, most of the glucose will be utilized and brain will get babaji ka tullu. That's why what will happen, liver will synthesize ketone body for brain, ketone body, starvation, ketoacidosis. And now I told you the sequence. Before moving further, in aims long time back this question was asked. Why I'm telling you, because they want you to remember the integration. Okay, this is cholesterol synthesis. Cholesterol synthesis occurs in cytosol, ketone body synthesis occurs in mitochondria, but initial two steps, but sorry, but initial two steps are same. Yes, these two steps are same, and this, the name of enzyme of this reaction is HMG coenzyme synthase. 2014 around, aims question was asked, enzyme common to cholesterol synthesis and ketone body synthesis, thiolase was not given in the option. The option was given HMG coenzyme synthase, and the answer was this. Okay, now I'm going a little bit deeper. It's really required. Conceptual question are asked. If a person goes into starvation, there is no glucose to produce energy, beta oxidation occur. Liver cell requirement of beta oxidation, by beta oxidation energy is produced, and the energy requirement of liver cell is fulfilled. Now liver cell will think of brain. If I don't synthesize, brain will die. So it do gluconeogenesis. It also synthesize ketone body synthesis. Suppose there is enzyme deficiency, medium chain acyl coenzyme dehydrogenase. What is medium acyl coenzyme dehydrogenase? Suppose this is palmitic acid. In palmitic acid we found that this first cut means palmitic acid 16 carbon, 16 carbon, this first cut is done by long chain dehydrogenase. Second cut is also done by long chain dehydrogenase. Further cut are done by means when 12 carbon fatty acid when 12 carbon fatty acid is left, further cut are done by medium chain acyl coenzyme dehydrogenase, medium chain acyl coenzyme. If this enzyme is deficient, then beta oxidation would not be complete. Liver cell will take one palmitic acid, cut Krebs cycle, cut Krebs cycle. Energy requirement of liver cell is not fulfilled. Previously, one palmitic acid was producing 8 acetyl coenzyme, but if medium chain acyl coenzyme is dehydrogenase is deficient, one palmitic acid will produce only 2 acetyl coenzyme. Energy requirement of liver cell will not would fulfilled. So liver cell will think of itself first. It will not divert this acetyl coenzyme to activate gluconeogenesis. So there would be no gluconeogenesis in medium chain acyl coenzyme dehydrogenase. There is deficiency, there is hypoglycemia. When this acetyl coenzyme is not present in sufficient amount, ketone body synthesis would not be there. In this patient, ketone bodies would be low. Clear? So medium chain acyl coenzyme dehydrogenase deficiency, there would be hypoglycemia, will lead to seizures, decrease ketone bodies. This is very important. Yes, why these decrease ketone body? Because less acetyl coenzyme produce, only 2 acetyl coenzyme produce from palmitic acid. Got the point? Now fatty acid, beta oxidation of fatty acid, beta oxidation of fatty acid. If odd chain fatty acid, the end product is, yes, if odd chain fatty acid, the last cut will be done the same, and 3 carbon compound fatty acid propionyl coenzyme. Question is asked, odd chain fatty acid beta oxidation occur, what is the end product? Propionyl coenzyme, which can take part in gluconeogenesis. You all know any compound having 3 carbon compound is a good substrate for gluconeogenesis, usually, not always. Okay, propionic acid is the fatty acid which can be used for gluconeogenesis, can be substrate for gluconeogenesis. Although fatty acids are not substrate for gluconeogenesis, acetyl coenzyme is not a substrate for gluconeogenesis. Acetyl coenzyme is activator of gluconeogenesis, not substrate. Point it out. Okay. Now I have already told you, when fatty acid is being synthesized, malonyl coenzyme is being synthesized at the same time. This beta oxidation of fatty acid will be kept in inhibited state, because fatty acid will not enter the mitochondria, because CPT1 is inhibited. CPT1 is inhibited by malonyl coenzyme. Yes, CPT1 is inhibited by malonyl coenzyme. So in fat state, beta oxidation is kept in inhibited state, one by insulin, second by inhibition of CPT1 by malonyl coenzyme. Questions are asked. Now, Refsum disease, yes, there is defect in phytanoyl coenzyme oxidase. Yes, that is phytanic acid, phytanic acid, odd chain fatty acid, branched chain fatty acid, chain fatty acid. And in this, yes, branch chain fatty acid, in this enzyme is deficient. Refsum disease, there is deficiency of alpha oxidation. Jamaican vomiting sickness, this fatty acid co dehydrogenase is deficient. These dehydrogenase are deficient. When dehydrogenase are deficient, you know Actually, it's not deficient. Jamaican vomiting sickness. Jamaica is a place in South Africa in which there is ackee fruit. Ackee fruit are having a compound hypoglycine. Hypoglycine. This compound inhibit this dehydrogenase. When this dehydrogenase is inhibited, beta oxidation don't occur. When beta oxidation don't occur, acetyl coenzyme not produced. Acetyl coenzyme is not produced, gluconeogenesis don't occur. Hypoglycemia, seizures. Hypoglycemia, seizures, decreased ketone body, Jamaican vomiting sickness. So most of time you will find wherever beta oxidation of fatty acid is inhibited, there is hypoglycemia, decreased ketone body. INICT 2022. Yes, Zellweger syndrome, defect in peroxisome. Yes, peroxisome is responsible for the beta oxidation of very long chain fatty acid C22, C22, very long chain fatty acid. This Zellweger syndrome is also called cyprohepatorenal syndrome. Okay, question was asked on peroxisome defect and peroxisome in Zellweger syndrome. Okay, a little bit faster. Beta oxidation of fatty acid and product is acetyl coenzyme, I told you, and product of old chain fatty acid propionyl coenzyme. Palmitic acid 16 carbon, just put 106 ATP. Energy from stearic acid 120. Yes, rare question but ask. Beta oxidation occur in mitochondria. Yes, ketone body synthesis occur in mitochondria. Gluconeogenesis start in mitochondria. Gluconeogenesis occur in both mitochondria and cytosol. Beta oxidation of very long chain fatty acid in peroxisome if defective, Zellweger syndrome. Starting compound is acyl, not acetyl. Fatty acid is also called acyl. Okay. Step is activation of fatty acid, 2 ATP used. Transport of fatty acid into mitochondria as acyl carnitine. I told you very important activator is carnitine. Yes. Every time when beta oxidation of fatty acid occur, acetyl coenzyme produced. Okay. So these are questions. Most of time they are asked. Now, this is again very important. Hyperlipoproteinemia, recently question asked. Remember, wherever you find triglyceride, triglyceride is having white color. Yes, it is white butter. Triglyceride is nothing but white butter. We know that chylomicron are metabolized by lipoprotein lipase. There is an enzyme lipoprotein lipase which is present at endothelium. This lipoprotein lipase metabolizes chylomicron, and activator of lipoprotein lipase is ApoC2. ApoC2 is activator of lipoprotein lipase. Question, question. Chylomicron is metabolized by lipoprotein lipase. Chylomicron contain mainly triglyceride. So if ApoC2 is defective, ApoC2 is defective, or lipoprotein lipase is deficient or decrease activity, then chylomicron will not be metabolized. Chylomicron will be increased, and chylomicron increase, that's why it is called familial hyperchylomicronemia. So familial hyperchylomicronemia or hyperlipoproteinemia type 1, apoC2 is defective, lipoprotein lipase defect is there, chylomicron is increased, also VLDL also increased, but chylomicron increases more than VLDL. Chylomicron and VLDL contain mainly triglyceride. Triglyceride is increased. I just told you triglyceride is nothing but white butter. I know that's a little bit, I'm not telling you the detail because it's rapid revision. In my regular class, I discuss it in detail, but for the time being we are summarizing depending on the questions which are being asked. You can solve very easily. White butter, wherever you find the triglycerides are increased, this is having white color, and question is asked, what would be the color of blood? In this case, blood color is red. Yes, cream of tomato soup appearance. Tomato soup is red, blood is red, cream is white, triglyceride is white. If blood increase triglyceride, the appearance would be cream of tomato soup appearance. First question. A question was asked in aims that there was a patient, you took sample, you found that milky white plasma. Milky white plasma is due to triglyceride. Milky white plasma. Milk word lactescent. Sometimes you will find lactescent blood, lactescent plasma. A question was asked, you took sample, you refrigerated the sample at 4 degrees centigrade overnight. In the morning you found that supernatant layer of white color. You know that butter floats over buttermilk. In the same way, triglyceride is very light, it floats over the blood, so it will make a superficial layer. If triglyceride is high, you know the color of triglyceride is white. And if triglyceride is so high, chylomicrons are so high in retinal blood vessels, you will find whitish color. Normally if you do fundoscopy, you will find reddish hue, pinkish hue. Yes, pinkish color, red color. But in patient having increased chylomicron, hyperchylomicronemia or type 1 hyperlipoproteinemia, the fundus would be whitish. That is called lipemia retinalis. White butter is granular, so mnemonic. It is not a concept, it's a mnemonic. If you deposit this white butter subcutaneously, it will appear like this. And this is called eruptive xanthoma. Eruptive xanthoma. Okay? Now, this is type 1 hyperlipoproteinemia, type 2 hyperlipoproteinemia, or familial hypercholesterolemia. You all know liver take LDL. LDL is having cholesterol. And we found that LDL is having ApoB100. This is ApoB100 which is present over LDL. Suppose this is LDL receptor. LDL receptor. Okay, so normally liver take LDL from peripheral blood vessel with the help of LDL receptor, and this LDL receptor bind with, again I repeat, LDL receptor bind with ApoB100. So sometimes we say LDL receptor recognize ApoB100, or ligand for ligand or ligand, whatever you pronounce, ligand for LDL receptor is ApoB100. And also ApoE, ligand for LDL receptor is ApoB100, and also ApoE. Question has been asked previously that ligand for LDL receptor both ApoB100 and ApoE. Got the point? Now this LDL is taken up by liver with the help of LDL receptor. If ApoB100 defective, LDL will not be taken, LDL will increase. LDL contain cholesterol, yes, familial hypercholesterolemia. If LDL receptor is defective, then familial hypercholesterolemia. We found there is a protein. Yes, this is really important. Recently asked question in INICT, question of pharma, medicine, whatever you say. Yes, there is a protein PCSK9. PCSK, PCSK9. This PCSK9 has property that it destroy this LDL receptor. It degrade this LDL receptor. If function of PCSK9 is increased, what I said, function of PCSK9 is increased. This is gain of function mutation. Gain of function mutation. If gain of function mutation, this LDL receptor will be destroyed. When LDL receptor will be destroyed, all LDL receptor are destroyed, LDL will not be taken from blood. Yes, LDL will increase cardiovascular disease. That's why we found that we can make antibody against PCSK9 to control hypercholesterolemia, and the antibodies against this evolocumab and alirocumab. Yes, antibody against. Okay, and whenever LDL is increased, cholesterol is increased. I usually remember cholesterol as egg yolk. And whenever cholesterol is increased, there is maybe chest pain, Yes, myocardial infarction, angina, yes, chest pain. So type 2 hyperlipidemia, patient present with chest pain. Type 1 hyperlipidemia, triglyceride are increased and triglyceride predisposed to acute abdomen pancreatitis, pancreatitis, surgery. Frequently asked question, increased triglyceride is predisposing factor for pancreatitis. Got the point? Yes. Now, PCSK9 antibody against it, evolocumab, alirocumab. Question recently asked question, if this cholesterol is deposited subcutaneously, it will give this appearance. This is called, yes, tendingenthoma, and you will find most common site is tendoachilles. Most common site is tendoachilles. Question is asked. We normally found that ApoE is responsible for the metabolism of remnant. Metabolism of VLDL remnant, yes, and chylomicron remnant. If ApoE is defective, then type 3 hyperlipidemia. Usually I write 3 like this. ApoE is defective. If ApoE is defective, then what will happen? This chylomicron remnant and VLDL remnant will not be deposited, will not be metabolized, will accumulate, and both triglyceride and cholesterol both increased. And when there would be increase of both, this is this will lead to palmar-xanthoma. Yes, this will lead to palmar-xanthoma in palmar crease. And tuberoeruptive xanthoma, cholesterol tuberous, triglyceride eruptive. So combination tuberoeruptive xanthoma. Sometimes it looks like grape-like appearance, bunch of grape-like appearance. Question is asked. And another name of type 3 hyperlipidemia is dysbeta lipoproteinemia. So in this question, you have seen that Parma-xanthoma is there. Yes, tubercular xanthoma is there. There is, yes, both triglyceride and cholesterol increased. This is familial dys-beta lipoproteinemia. One more question. This frequently asked question, 2-3 times this has been asked. Tangier disease. You must have taken tang, rasna, the orange color. Yes, the most common flavor of rasna is orange. Most common flavor of tang is orange. Orange color tonsil. If you are drinking rasna, tang again and again, your tonsil would become orange color. So Tangier disease, orange color tonsil, typical. Okay, and what is the color of cholesterol? Yes, orange. Color of cholesterol is orange. So you can say there is defect in the metabolism of cholesterol, and in this ABCA1 gene is defective, so there is no formation of HDL. HDL will be decreased, means HDL will not be able to take cholesterol from peripheral tissue. Cholesterol will deposit in peripheral tissue. So you can easily understand this is Tangier disease. This question you can easily solve. 7-year-old boy presented with severe abdominal pain, acute abdomen. Acute abdomen may be due to acute pancreatitis. On examination, he had xanthomas. Usually, whenever triglyceride is increased, eruptive xanthomas are there. They have not mentioned, but you will remember. Blood sample was taken for workup. Blood sample had milky appearance of plasma. Yes, you take blood sample, milky plasma, cream of tomato soup appearance. You took the sample and refrigerated at 4 degrees centigrade overnight, supernatant layer of white color, lactescent blood. Triglycerides are increased, chylomicrons are increased. So in this question, which live protein is increased? Chylomicron is increased. Now, in this question, laboratory investigation in patient, LDL was very high. LDL is having bad cholesterol, means that is hypercholesterolemia. But the LDL receptor was normal. If LDL receptor is normal, the defect may be ApoB100, I told you that in familial hypercholesterolemia, defect may be LDL receptor, defect may be ApoB100, defect may be PCSK9. This is very specific mutation, gain of function mutation. Most of time you will find whenever mutation is occurring, the function of protein is decreased. But in this PCSK9, function is increased. That lead to hypercholesterolemia. Yes, that's why we make antibody against PCSK9. Two times question has been asked. Evolocumab, alurocumab. So in this, if LDL is high, LDL receptors are normal, there may be defect in the ApoB100. So answer is C, ApoB100 mutation. Now we come to ketone body utilization. You all know three compounds which are termed as ketone bodies: acetoacetate, beta hydroxybutyrate, and acetone. This is ketone body utilization. Acetoacetate react with succinyl coenzyme with the help of enzyme thiophorase. Thiophorase. This enzyme convert acetoacetate to acetoacetyl coenzyme. Then it is being utilized by different cells, not all cell in our body, only important organs, most vital organ, brain, utilize ketone body in starvation. Yes, cardiac muscle, heart, utilize ketone body in starvation. Skeletal muscle also utilize ketone body. It is also essential. Even when you are sleeping, some muscles are doing work. So nature is very wise. Nature thought that if I make glucose by liver, gluconeogenesis, then this glucose will be utilized by different cells. If I make a specific compound which can be utilized by important organs in case of starvation, that will be utilized by only and only important tissue which are very essential for survival. That's why brain utilize ketone body, cardiac muscle, heart utilize ketone body, stated muscle. And cardiac muscle utilize fatty acid even in fat state. This question is asked. Fatty acid even in fat state means when you are having sufficient amount of glucose, even then our heart utilizing fatty acid. You have heart, दिल तो पागल है, दिल दीवाना है, क्यों है पागल? Because it utilizes fatty acid even in fat state. Liver is the main organ of metabolism. It most of the pathway occur in liver, but liver is the organ which can synthesize ketone body but can't utilize due to deficiency of can't utilize due to deficiency of thiophorase. This question is asked. Liver is the organ, so answer is liver. Type 1 hyperlipoproteinemia, hyperchylomicronemia. I told you lipoprotein lipase or ApoC2 is defective. Type 2 hyperlipidemia or hypercholesterolemia. LDL receptor may be defective. ApoB100 may be defective. PCSK9 may be defective. Gain of function mutation. LDL receptor adapter protein may be defective. Leave that. Type 3 hyperlipidemia, ApoE is defective. Another name is beta lipidemia. I told you in this cholesterol and triglyceride both are increased and there is tuberoreptive xanthoma, palmaxanthoma. Now protein. You all know proteins are made up of amino acid. There are 20 amino acid which usually take part in protein synthesis. But 21st amino acid is selenocysteine. So normally 20 amino acid take part in protein synthesis. Suppose I give you a protein having two amino acid. One is alanine, then another second is valine. Suppose, and third is leucine. I'd ask you, what is the structure of this protein? What is the primary structure of this protein? You will say, sir, in this protein there are three amino acid. One is alanine, second is valine, third is leucine. Means you told me the amino acid sequence of this protein. I will say, okay, this is the primary structure of protein, and we all know amino acid have common structure. Yes, amino acid is having common structure, and whenever two amino acid join, they form amide bond. Which bond? That this CONH. This is amide bond. Amide CONH. And we say in protein, amino acids are joined by peptide bond. So peptide bond is nothing but amide bond. Chemically, peptide bonds are amide bond. Amide bonds are covalent bond. And if you want to cut this amide bond, you must add water. You must add water, and you will find that there will be formation of acid and amine. And we know that any enzyme which cut a bond with addition of water is classified as hydrolase. Again, I repeat, any enzyme which cut a bond with addition of water is classified as hydrolase. So all proteolytic enzyme cut this amide bond, cut this peptide bond. So all proteolytic enzyme cut this peptide bond with addition of water. So that's why all proteolytic enzymes are hydrolase. Question is asked, pepsin is hydrolase, trypsin is hydrolase. Okay. All digestive enzymes are hydrolase. Mnemonic. All digestive enzymes are hydrolase. Yes, Baba Ramdev said, yes, if you want to digest something, take water after food, because any enzyme which will digest will require water. So all digestive enzymes are hydrolase. Okay. Trypsin is hydrolase, amylase is hydrolase, lipase is hydrolase. So now you will remember. I come to my main topic. Primary structure is stabilized by covalent bond. And suppose I give you a protein having 1000 amino acid. These 1000 amino acid will not remain in a linear chain. They may fold. You know, protein are globular in shape. So these protein molecule, so these amino acid fold themselves. So sequence of amino acid may fold themselves in a helical structure, then it is called alpha helix. It may fold. It may fold in a plane like structure, just like is this paper. Yes, just like this paper, that is called beta sheet. That secondary structure is alpha helix or beta sheet. Question is asked, secondary structure is stabilized by hydrogen bond. There is one more question, that alpha helix. Proline is never found in alpha helix. What I said? Proline is never found in alpha helix. Glycine is usually not found in alpha helix. If question is asked, which of the following amino acid is not found in alpha helix? Best answer would be proline. Okay. And these two amino acid are found in beta band or beta turn. Again, question, recently asked question. So this is secondary structure. Tertiary structure. Suppose a protein want to do function. Suppose this is a protein, it want to do function, it want to carry this pen. Although it's very heavy, it can't carry. Yes, but if I fold, if I fold this protein randomly, this is tertiary structure. This is tertiary structure. Now it want to carry this pen, so heavy pen it can carry. Yes, this is tertiary structure, can do function. And tertiary structure is stabilized by disulfide bond, hydrophobic hydrogen ionic bond. Main bond is disulfide bond. Question is asked, protein acquire function at tertiary level. Quaternary structure means more than one functional polypeptide chain, just like hemoglobin. Four globin chain are there. Four globin chain are there, and each globin chain can do function. More than one functional polypeptide chain, quaternary structure. Myoglobin is having only one polypeptide chain, functional polypeptide chain. So myoglobin is having tertiary structure but not quaternary. Quaternary structure is mainly stabilized by hydrophobic bond. We found if you heat a protein, this protein is denatured. Denatured means secondary, tertiary, quaternary structure are lost. And if secondary, tertiary, quaternary structure are lost, protein can't do function. Protein can't do function. Okay, denaturation. One question, primary structure is best detected by mass spectrometry. Question. Another technique is advanced technique. Mass spectrometry mnemonic. Mass means molecular weight. This technique, mass spectrometry, can tell you the amino acid sequence in a protein, can also tell the molecular weight of protein. Question is asked, which of the following technique can determine amino acid sequence in a protein and also molecular weight of protein? The answer was mass spectrometry. Secondary, tertiary, quaternary structure best determined by X-ray crystallography. Latest edition of Harper, yes, cryo-electron microscopy. 32nd edition of Harper, cryo-electron microscopy has replaced this X-ray diffraction crystallography. So this may be a potential question. Okay, another technique, NMR spectrometry. So this is about structure of protein. Questions are asked. Now you know branch chain amino acid, three branch chain amino acid. Mnemonic is live. L leucine, L for leucine, yes, I for isoleucine, V for valine. Leucine, isoleucine, and valine, these are three amino acid, yes, which have branch chain. Side chain is branched. Okay, and if this branch chain amino acid are not metabolized, normally they are metabolized by branch chain, branch chain alpha keto acid dehydrogenase, and this enzyme catalyze oxidative decarboxylation. Oxidative decarboxylation, remember, decarboxylation cofactor is simple decarboxylation. I write here simple decarboxylation, simple decarboxylation cofactor, yes, repeat, B6. Oxidative decarboxylation of alpha keto acid, B1. Cofactor for carboxylation, for carboxylation, joining of carbon dioxide, that is biotin. All three question. Okay, simple many times they ask question like this: histidine is converted to histamine by decarboxylation. You must know histidine to histamine is simple decarboxylation. So three four example of simple decarboxylation you must mug up. Histidine to histamine, cofactor would be B6. Yes, tryptophan to serotonin, tryptophan to serotonin, tryptophan to serotonin. Simple decarboxylation cofactor is B6. Glutamate to GABA. Simple decarboxylation cofactor is B6. All other decarboxylation you will find cofactor is B1. You will remember like this. Pyruvate to acetyl coenzyme B1. Yes. Alpha ketoglutarate to succinyl coenzyme B1. Yes, carboxylase biotin. Got the point? So oxidative decarboxylation I discussed. Branch chain alpha keto acid dehydrogenase is deficient. It is called maple syrup urine disease. Recently asked question, multiple time question is asked. In maple syrup urine disease, this enzyme is deficient. Which amino acids are not metabolized? Live, LIVE, leucine, isoleucine, valine. Clear? They accumulate. Recently INICT 22, as repeat questions are being asked, topics are being asked, so focus on the repeat question definitely. Yes, you all know basic amino acid. These are three: histidine, arginine, and lysine. Whenever you find the word basic, basic means having positive charge. We found that histidine is least basic and it is having only one positive charge. I am quantifying myself so that you may remember and you may solve question easily. Yes, histidine is having only one positive charge, arginine is having three positive charge, lysine two positive charge, means maximum positive charge on arginine. So most basic is arginine, least basic is histidine. How to remember? Yes, one positive charge, he is, his, he is rm. English grammar we have read, he is, एक ही होगा बेचारा. There are so many charge on arginine, multiple positive charge. So many charge on arginine, multiple positive charge. Okay, so histidine is having one positive charge, least basic, arginine, maximum positive charge, lysine. Yes, you know, these are basic amino acid. Question is asked that most basic is arginine. And you know, if you do electrophoresis, this is cathode, this is anode. Yes, what is the charge on cathode? Yes, many times we do remember, so hard work we do and ultimately we forget. Yes, cathode is having negative charge, anode is having positive charge. Don't get confused anode as anion. Anode is having positive charge, cathode is having negative charge. Question is asked, if we do electrophoresis, which amino acid will move fastest towards cathode? They will give you three four options, and they will give one option arginine. Cathode is having negative charge. On electrophoresis, arginine is having maximum positive charge, so arginine will move fastest towards cathode. So they will give you electrophotogram picture like this, and they will ask, this amino acid is definitely maximum positive charge, arginine would be here. Yes, because opposite charge. So electrophoresis movement depends on, yes, electrophoresis movement depends on charge, size and shape. Yes, all these three. Yes, main force is charge, but movement in electrophoresis also determined by shape and size. Yes, you will remember this. Now, aspartic amino acid is having negative charge, glutamic acid having negative charge, so they will move fastest towards anode. Okay, so these are five amino acid. They are polar with charge. Point it out. They are polar with charge. Okay. Figlu is a metabolite of histidine. Yes, histidine is metabolized to figlu. Yes, figlu form glutamate, and this figlu is converted to glutamate with the help of folic acid, with the help of what I said, folic acid or tetrahydrofolate. In this reaction, tetrahydrofolate is converted to form amino-tetrahydrofolate. If folic acid is deficient, then figlu will not be converted to glutamate. Figlu will be excreted in urine. Figlu test positive. If question is asked, figlu is a metabolite of, answer is histidine. But question is asked, figlu test is done for. Figlu test is done for folic acid deficiency. Both are question. Figlu test done for folic acid deficiency. Figlu is a metabolite of histidine. Aromatic amino acid, you all know, phenylalanine. Phenylalanine, this is phenylalanine. If you hydroxylate it, if you join OH group to phenylalanine, it becomes tyrosine. OH means O. I usually say O looks like tire. O looks like tire. So this is become tyrosine by phenylalanine hydroxylase. If phenylalanine hydroxylase is deficient, then it is phenylketonuria. Okay, in this mousy odor or musty odor. Okay, due to accumulation of metabolite of phenylalanine. Phenylalanine will accumulate. Phenylalanine will cause blood-brain barrier. Patient will develop mental retardation. Sometimes you will find the child, infant, or you can say neonate is having very large amount of vomiting, or intensity of vomiting is very high, and such a intractable vomiting you will find, you may confuse with pyloric stenosis. What I said, pyloric stenosis. Yes, vomiting is there. Yes, this is phenylketonuria, phenylalanine hydroxylase deficient. And if this is deficient, tyrosine will not be synthesized. Normally in our body, tyrosine is synthesized from phenylalanine. But if this enzyme is deficient, tyrosine will not be synthesized. Tyrosine must come from diet. Question was asked that phenylketonuria, tyrosine becomes essential amino acid. Becomes essential amino acid. Normally tyrosine is oxidized. What I said? Oxidized. There is an enzyme tyrosinase. Tyrosinase. If you burn a tire, you will find which type of pigment? Black pigment. Black pigment is melanin. Black pigment is melanin. Normally, tyrosine is burned by tyrosinase to form melanin, and the cofactor is copper. Yes, if you burn a tire, you will find black, uh, you know, uh, residue, and the wire would be reddish, just like copper. Yes, it's my mnemonic. You can, yes, the copper is cofactor for tyrosinase. Most of the oxidation, copper is cofactor. Many times you can use this concept. Tyrosinase. Yes, if this enzyme is deficient, melanin will not be produced. That is albinism. Albinism. Okay, normally tyrosine is converted to dopamine, norepinephrine, epinephrine. These are catecholamines. Question is asked, tyrosine is precursor for catecholamines. Question is asked, tyrosine precursor for thyroid hormone. Yes, tyrosine is also metabolized to homogentisate. Homogentisate is converted to mallyl acetoacetate by homogentisate oxidase. Homogentisate oxidase or homogentisate dioxygenase. If this enzyme is deficient, then it is L-captonuria. 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 Yes, urine turns black on standing. L-captonuria. If you read like this, Ka-la, Ka-la. Opposite, Ka-la. Urine turns black on standing. Okay, this is frequently asked question. L-captonuria, phenylketonuria, albinism questions are asked. Now, you must have an idea. This is recently asked question. So many question. Amino acid. Yes, I know it's really difficult to remember structure of each and every amino acid, but for the time being, yes, we can remember this is methionine. Even if you don't remember, it's okay, at least have idea this is methionine having methyl group attached with sulfur. If I draw it again with little bit modification, CCSH, this is homocysteine. This is homocysteine. Okay, even if you don't remember the structure, I say at least you must know there is a difference between homocysteine and methionine. The only difference is methyl. This is methionine having methyl group. So homocysteine and methionine, the only difference is methyl group. So normally in our body, homocysteine is converted to methionine by enzyme homocysteine methyl transferase. This homocysteine methyl transferase will transfer methyl group to homocysteine. When you transfer methyl group to homocysteine, it will be converted to methionine. And we found that if this enzyme, sorry, if this enzyme cofactor is B12 and tetrahydrofolate or folic acid, if any one of these is deficient, if these B12 or folic acid is deficient, this enzyme will not work. Homocysteine will not be converted to methionine. Homocysteine will increase. Means this is called non-classical homocysteinemia. This question is asked many times. If B12 is deficient, there is megaloblastic anemia. You all know. Folic acid deficiency, megaloblastic anemia. A patient came to me having megaloblastic anemia. I don't know this megaloblastic anemia is due to folic acid or B12 deficiency. I thought that I can assess by knowing the homocysteine level, but you know B12 and folic acid deficiency both lead to homocysteinemia. So patient presented to you having megaloblastic anemia, homocysteinemia, both B12 and folic acid may be deficient. How you will differentiate? Yes, you will say, "Oh, sir, we will give both of B12 and folic acid to the patient." No, no, dear. You must be precise in question, at least for question, not for clinical practice. I know. Yes, if this patient came to you, I will check methyl malonyl level. Methyl malonyl coenzyme A is normally converted to succinyl coenzyme A by mutase, and the cofactor is, yes, please do reply. Oh, sorry, it's not live class. It is B12. If B12 is deficient, then there would be methyl malonylic acid urea, increase methyl malonyl. So in this patient, what I will do, I will check increase methyl malonyl level. If it is increased, this is B12 deficiency. It is normal. It is folic acid deficiency. You will differentiate like this. If cystathione beta synthase, cystathione beta synthase, what is cystathione beta synthase doing? Homocysteine, normally in our body, homocysteine bind with serine with the help of this enzyme. Cofactor for this enzyme is B6. Question. Cystathione beta synthase cofactor is B6. If this is deficient, this enzyme is not working, then this is called classical homocysteinemia. Means normally our body convert homocysteine to, yes, cysteine by multiple steps. Yes, we are not concerned. Yes, But if this enzyme is deficient, homocysteine will not be metabolized. There would be homocysteinemia, and in this cysteine will not be synthesized. Normally in our body, methionine is converted to normally in our body methionine is converted to cysteine. Methionine is essential, cysteine is non-essential. But if cystathionine beta synthase is not there, not working, cysteine will not be synthesized. Cysteine will become essential. Essential means it must be supplied in diet. This question INICT recently, that which compound you will give in the treatment, and option, relevant option was given cysteine. The answer was cysteine. Got the point? Now you all know there are so many adverse condition occurring in our body. Fever is there, temperature increases, shock like situation is there. So we found that our nature has protected us, because you know that protein acquire function at a tertiary level. Yes, if a protein want to do function, I told you, if a protein want to do function, it must have at least tertiary level structure. Means a proper folding must be there. But I told you, if you heat a protein, this protein is denatured. If this protein is denatured, it can't work. Yes, so we found that in our body we have chaperone, चेपी, जैसे पतंग उड़ाते हैं ना, चेपी लगाते हैं. तो if patient, suppose a cell is having a protein, temperature is increased, adverse conditions, so shock like condition occur, protein is denatured, protein can't do function. Now what you will do, what cell will do? There are two options. If protein is there, this protein is denatured, this protein is denatured, I have two options: either degrade it or correct it. There is a concept, or there is a protein known chaperone. Chaperone, another name is heat shock protein. This heat shock protein comes and try to fold this protein, just like a villain in movie, yes, in jail, try to threaten hero by folding an iron rod like this, use energy. In the same way chaperone use energy. Question: chaperone use energy and it fold protein again normally. Yes, if chaperone is able to do proper folding, this cell, this protein will do work. But if chaperone is unable to fold this protein again, what it will do? It will leave this protein as such? No. When chaperone is unable to fold it correctly, chaperone will attach ubiquitin. What I said? Ubiquitin. This ubiquitin is a stamp that I tried my best but could not repair. Better to degrade this protein. Now this misfolded protein will go to proteasome and will be degraded. Okay, got the point? So proteasome degrade those protein which have ubiquitin. Got the point? Yes. So benefit of this, that proteasome will recognize, okay, chaperone has tried to fold this protein properly, could not fold it properly, better to degrade. Now this question: cystic fibrosis, there is a phenylalanine mutation. Recently asked question: phenylalanine mutation occur. Yes, CFTR protein fold incorrectly. Patient cell modify this abnormal protein by attaching ubiquitin molecule. What does it mean? If ubiquitin is attached, means cell could not repair it. If it is not unable to repair, it attach ubiquitin. Now it is degraded with the help of proteasome. Answer is it will be degraded by proteasome. 2-3 time question. Yes, most accepted method of HbA1c estimation is ion exchange chromatography. Yes, these three questions were asked from this topic. I can't skip this topic untouched. Yes, so and there are so many topics in MLE pattern, so you can't afford to forget this structure. I know it's difficult, but you all know pure as gold, pure purine, pure as gold. Yes, you all know purine bases are adenine and guanine. Purine bases adenine and guanine. This is adenine and guanine, and purine base are having two rings. How many rings are there? Two rings are there. Purine base are having two rings. One, two. one, two, one, two. Pyrimidine, you are having only one ring, means one ring is cut. One ring is cut. Mnemonic is again cut. C-U-T, cut. So three pyrimidine base, two purine base, on which questions are frequently asked. Now suppose a structure is given to you and you found two rings having nitrogenous base are there. Immediately you will search for amino group. Immediately you will search for amino group. It is mnemonic. If you find only amino group in side chain, A for amine, A for adenine, if you find amine group plus additional keto group, then it is guanine. Previously I used to teach that if you see amino group at the top, A for amine, A for adenine, but later on I found in many MLE pattern question they rotated the structure, so not on top. Simply remember adenine only amine, guanine amine plus keto group. Got the point? Now, cytosine, uracil, and thymine recently asked question. Very difficult question it was. Yes, let me tell you. Yes, this is cytosine. Amine group is there and keto group is there. And we found that in our body, whenever amino group is removed, there is formation of keto group. If I remove amino group from cytosine, it will be converted to keto group, and cytosine will be converted to uracil by an enzyme present in our body. The name of enzyme is cytosine deaminase. Cytosine deaminase. So you can say cytosine and uracil only differences of amino group. Amino group can be converted to keto group by cytosine deaminase. We found that uracil and thymine only difference is methyl. Methyl. Thymine is having extra methyl group, so uracil can be converted to thymine by methylation, and this methyl group come from folic acid, tetrahydrofolate. So methyl group donor is folic acid. This question was asked recently in a different way. Two three questions are there recently asked question. Recall based question. Yes, due to deficiency of folic acid, what will happen? You all know DNA is having ATGC, RNA is having AUGC. What is the difference between T and U? T and U, the only difference is of methyl group. If folic acid is not there, then uracil will not be converted to thymine, because folic acid act as methyl group donor. So if folic acid is not there, then DNA will have uracil in place of thymine. So there was a question that due to which compound deficiency there would be insertion of uracil in place of thymine. Yes, answer would be yes, folic acid deficiency. So one question. Second question, simple question. Uracil is coming to thymine. Methyl group donor is yes. Methyl group donor is folic acid. Third question which was asked, yes, now you can easily see that cytosine, if you deaminate cytosine, it will be converted to uracil. Yes, and if you attach methyl group to it, it becomes thymine. What was the question? Really difficult question, but was asked. Yes, they asked deamination followed by methylation of which base give rise to thymine. Again, I repeat, deamination followed by methylation of which base give rise to thymine. The answer was cytosine. Got the point? What is the structure indicated below? What is the structure indicated below? Yes, you know base plus sugar nucleoside. If I say this base, two rings are there. Two rings, whenever you will think a purine base, it would be adenine or guanine. Only amine adenine. Base plus sugar nucleoside. This is purine nucleoside, I know, and this is adenosine. Adenine plus sugar, adenosine. So this is option E. Adenosine is the best answer. But adenosine, if not given in the question, you will say it is purine nucleoside. Base plus sugar nucleoside. Okay, got the point? Yes, misincorporation of uracil in DNA seen in deficiency of. Yes, this was the question, recall question. Okay, yes. Now, this is pentose sugar, C1, C2, C3, C4, C5. Ribose sugar is a pentose sugar. If I draw, this is HOH, C2 is having HOH, C3 is having HOH, C5 is having HOH. So ribose sugar is having 5 carbon, 4 OH, and if I remove one oxygen from C2, removal of oxygen is called deoxygenation. Ribose, this is ribose sugar. You removed one oxygen. Removal of oxygen is called deoxygenation. Ribose becomes deoxyribose. If I remove one more oxygen from C3, why I'm so much concerned about this difficult topic? Recently, INICT from this page, 3 questions were asked. 2 questions I have already discussed. Third, I am going to discuss. It will take little bit time, but trust me, it's again potential topic. So many times in MLE, this question is asked. Okay. So this is deoxyribose. If oxygen is removed from C2, deoxyribose. If I remove oxygen from C2 and C3 both, then it become dideoxyribose. So if I draw like this, yes, this is C1, C2, C3, C4, C5 ribose sugar. Sugar bind with base, base plus sugar nucleoside. Suppose this is adenine, and C3 is having HOH, C5 is having HOH. I told you pentose sugar, ribose sugar is having 4 OH. At this time, I am mentioning OH group at C5 and only C3. I am mentioning OH group at C5 and C3. Again, I draw one more pentose sugar, C1, C2, C3, C4, C5.